Okay, so my name is Caroline Kou. I'm a pediatric immunologist at UCLA. So thank you very much for having me here today. I've been working on on alternative therapy for X-linked hyper-IgM for the last 5 years. So I'm always very excited to be able to share with you some of my work. And so because we're a small group and because it can be a little bit complicated, you're welcome to raise your hand and stop me at any point if something doesn't make sense. So I am working on, instead of saying gene therapy, I'm calling it gene editing, and I'll explain to you why we're doing this for X-linked hyper-IgM in a second. And so just, a little bit of background. So primary immune deficiencies are a really large group of chronic disorders which are due to the dysfunction of the body's immune system. And so patients will present with recurrent infections of multiple organs, such as the skin, the respiratory tract, the brain, the GI tract, the blood. And it can really vary from people being asymptomatic and generally healthy to incompatible with life or life-threatening. And so if we kind of look at where the immune cells come from, we understand where our treatment options are coming from. So this is the hematopoietic tree. And so at the very top is a multipotent stem cell. And so this stem cell lives in the bone marrow, and it gives rise to all of the cells in your blood. And so I'll point out kind of the major immune cells. There's many more, but so here's the main cell, and there's B cells and T cells that Maite talked about, and which can be affected in hyper-IgM, as well as a lot of other immune cells like NK cells, monocytes, neutrophils. Oops. And so if you have a disease in any of these cells, kind of in the bottom, you could potentially cure it by replacing that cell at the very top, which is that multipotent stem cell. And what we've been doing is they're called allogeneic bone marrow transplants, which is taking normal bone marrow from a healthy donor and giving it to somebody who has a disease in any of these kind of downstream cells. And that way, for the rest of the life of this affected individual, they'll always produce normal now B cells, T cells, and all other cells in the bloodstream. And so we call that an allogeneic bone marrow transplant because you're getting the bone marrow or the stem cells from a donor that is not the patient themselves, so from somebody else. And so we've been doing this for a lot of different primary immunodeficiencies since the 1960s. Some of them are listed here, but really the list is much, much longer now. But one of the major limitations of allogeneic transplant is the unwanted immune response between the patient and the transplanted cells. So two main things can happen. One is that the patient can reject the cells that they're receiving, or the other thing can happen is that the cell, there may be some T cells that come with the donated cells, and that can attack the patient. And so we're often having to deal with this kind of yin and yang of rejection and graft versus host disease, which is cells from the donor attacking the patient. And so because of these complications, and also because not every patient has an HLA matched donor, was born the idea of gene therapy with autologous transplant. So autologous means that these stem cells are coming from yourself or from an affected individual. And the way that works is if you know what gene is diseased and is causing a disease, and if you can take that normal gene and introduce it into a stem cell, which is not only multipotent, meaning giving rise to all these different cells in the blood, but also self-renewing and everlasting, then you could also cure these diseases while avoiding a lot of the pitfalls of a normal transplant. And so you imagine that if we can introduce this normal gene into a patient's own stem cells, that this normal gene would then be passed down to all of its daughter cells and all of the cells that are in the blood. Okay, so for X-linked hyper-IgM, if you have a disease, if your problem is in the T cells, then that would now have the normal gene. And so the way that this practically would look, and this is what we've been doing in clinical trials for other primary immune deficiencies, is that you would harvest these stem cells from an affected individual, either as a bone marrow harvest or as a peripheral blood stem cell harvest. And then you would take those stem cells into the lab and you would purify them in the lab. And then after you purify for the most pure stem cells, you would introduce the normal gene into those stem cells. And that takes about 2 to 3 days. So during that 2 to 3 day process where you're processing these stem cells and introducing the normal gene, the patient would be in the hospital and they would get a small dose of some sort of cytoreductive agent. That is it's like a chemotherapy, which is at a lower dose and basically just clears a little bit of room in their bone marrow so that when you give them back their stem cells, they have somewhere to go and reside. And so after that 2 to 3 day period, then they are given back these gene-corrected stem cells, and it looks just like a blood transfusion. Okay, and then we can follow them for years looking at whether or not the gene is expressing properly and looking at safety. And so the way that we have been delivering a normal copy of the gene is using viral vectors. And the reason we do that is because viruses, especially the HIV virus, naturally infects you because it is able to get into your cell. It actually is able to get its own DNA and integrate it into your DNA. So it kind of escapes your own immune surveillance. And so we've taken kind of that backbone and modified it so we take away all the bad parts about HIV. And then we make it carry the corrective gene that we want it to carry. So it's now pictured in that kind of blue curly thing. So if that's, the gene that we want to introduce into a stem cell, we take the stem cell, we incubate it, or we kind of mix it together with this viral vector. And the viral vector has its own machinery to enter the cell. It kind of opens its capsule, and it goes through some DNA processing. And that DNA, after it's processed, can enter the nucleus, where your own DNA is, and it integrates into your natural DNA, okay? And once it does that, it can now express the protein or whatever is missing therapeutically, and it's stably transmitted to all of its daughter cells and also to others, when it's regenerating, when it's giving rise to its own stem cells. So this is considered curative. And so this is kind of what it would look like. So the dotted squiggly lines are imagine if you're the patient, that's your own natural DNA. So you have a ton of it, everywhere. And we're able to use this viral vector and we introduce this therapeutic gene, and that integrates randomly into your genome. So it's shown here kind of trapped somewhere between, a part of your DNA. And so that therapeutic or corrective DNA will remain a stable part of the cell's chromosome, and it gets passed on to all of its daughter cells. But this form of gene therapy is not perfect. It works really well, but it's not perfect for two big reasons. One is that the expression of the gene is not normally regulated. So in all the thousands of genes that you have, you can think of it as having light switches. So naturally you have it's telling this gene turn on now or turn off now or stay on all the time or stay off most of the time but only turn on when you're sick, okay? But when you do traditional gene therapy, there isn't anything that's telling the gene to turn on or off. It's just there. And so what we need to do is basically add this artificial light switch that tells the gene to always be on. And so you might imagine that maybe most of the time that's okay, but some genes are not meant to always be turned on, okay, not always expressed. The other thing is that this gene that's supposed to be therapeutic and that we're adding can integrate randomly, and it can integrate near a proto-oncogene and accidentally turn on an oncogene and cause cancer. And so the way I've shown that is, remember that on-off switch that we have to add to express the therapeutic gene? Well, what if that on-off switch integrates too closely to a proto-oncogene? And then now you'll have cancer because that gene is now always turned on by what you've added in. And so we know, based on previous work, that traditional methods of gene therapy does not work for X-linked hyper-IgM. So these are two publications that came out, and they treated mice with X-linked hyper-IgM using the traditional form of gene therapy. And so the good thing was they found that they were actually able to fix the immune system in the majority of these mice, which was good, meaning gene therapy is possible. But the bad thing is that the majority of the mice in both of the studies developed cancer. And it's probably because CD40 ligand needs to be tightly regulated. So in our bodies, CD40 ligand expression is quite low. It's when we're well and healthy, it's not turned on. And it's only turned on when we are exposed to a bacteria or virus and we need to react to it. So we know that traditional forms of gene therapy cannot work for X-linked hyper IgM. So the alternative comes to us in the form of what we call programmable nucleases. And so these are the main players that we've been using. I'm going to focus on the bottom two, called TALENs and CRISPR-Cas9. And CRISPR-Cas9 has been in the news a lot, so you may have even read it like in Time and a lot of LA Times, wherever you are in newspapers a lot, because it's really revolutionized the way that we do gene editing, because it's so easy to manipulate in the lab and it's so easy to deliver to cells. Okay, so I'm going to talk to you first about TALENs, because that's what I started working with. That's what was available when I started this project. And then also about CRISPR-Cas9, because that field blossomed and then I started using that as well. And so just a little bit about how these work. So these nucleases break DNA. Okay, so breaking DNA is really not great. You don't want to do that on a regular basis. But we break the DNA and we utilize the cell's natural repair mechanism to do what we want. So what happens when your DNA gets broken? This happens all the time when you're exposed to UV, when you get, radiation, all the time. And so two main repair pathways can happen. One is called non-homologous end joining. So if you imagine your double-stranded DNA gets broken, this is it's like a Band-Aid. So it's like, let's quickly stick this back together before any other parts of your DNA get lost or get mutated. So they just quickly stick it back together. This is okay because then you won't have additional damage with a double-stranded DNA break. But it's not perfect, because if you imagine you've now broken the DNA, you quickly stick it together, you've lost some pieces, you may have accidentally added another little piece of DNA. So it generally will disrupt the gene. Okay, so this isn't great, but it's good for not allowing more damage to occur. The other main pathway that can occur is called homologous recombination, and this is what we want to happen for gene editing. So in certain phases of your cell cycle, especially when your cells are replicating, at some point it'll have an identical copy of DNA before it splits off into being a second cell when it's replicating. If you happen to have a break in your DNA at that point, it could use that identical copy as a template. So kind of take that over, cross it over, and use it as a repair template, and now it's repaired itself in a sequence-perfect manner. Okay, so now not only have you repaired this break, but your gene remains functional because you haven't added or lost any DNA base pairs. And so that is really what we want to do for gene editing for X-linked hyper IgM. So this is kind of a cartoon depiction of what happens. I call it gene surgery, because that's essentially what we're doing, is we take TALENs or CRISPRs, we introduce them into a patient's cells. Those TALENs or CRISPR-Cas9 will cause a break in the DNA exactly where we want it to. In this case, it's at the CD40 ligand gene. If we don't do anything, if we just allow it to have the CRISPR and we let it break, then this non-homologous end joining will occur and will disrupt the gene. However, what if instead of having a sister, instead of having the sister chromatid when it's replicating, what if we artificially provide it with the corrective donor that it can use as a repair template? And so what's being shown here is this blue part is the therapeutic gene, so it would be the CD40 ligand gene. And this purple part and this purple part, it matches this purple part and this purple part right around the cut site. So now the gene thinks that, oh, I have something that I can use to repair myself. So it'll find that, and then you kind of trick it into incorporating the corrective blue sequence that's in between. Okay, so that's our goal for gene editing. And then so now we'll kind of go into more hyper IgM. This is now review because Maite has reviewed this with you already. But the hyper IgM syndromes are characterized by the inability to class switch, right? So as was presented earlier, you only make IgM and you can't switch to IgG, A, or E. And the reason for that is because in order for us to class switch, our B cells and our T cells have to communicate. And it communicates through various routes, but one absolute requirement is communication between CD40, which is always on and expressed in B cells, and CD40 ligand, which is expressed by T cells, but only when the immune system is activated. So if you lack CD40 ligand, you have the X-linked form of disease, while when you lack CD40, you have autosomal recessive, as well as the AID and the UNG that was mentioned earlier in the B cell. And so patients with X-linked hyper IgM, they have many recurrent infections. A lot of them are invasive, this was presented earlier, in addition to autoimmunity and also increased risk of cancers, because we know that the immune system is important for immune surveillance, so looking for cancers and clearing them. And so this is also from Maite's recent paper, but despite supportive care with immunoglobulin, prophylactic antibiotics, and with transplant, we know that survival probability is still, at least from the data that we have, is still not great long-term for affected patients. And currently, the only known cure is hematopoietic stem cell transplant, but there's many different concerns, such as reactivation of chronic infections, worsening of pre-existing conditions like lung damage, graft versus host disease, which is where the donor cells can attack the recipient cells, and also because this is a very rare disease and there's not one center that's an expert in this. When to time the transplant and how to condition them is pretty different at different institutions. So there's really no kind of centralized treatment center for this disease. If we look at the gene that's responsible for X-linked hyper IgM, it's CD40 ligand. And this is a picture of, it's a little bit old, but it's showing all the disease-causing mutations in CD40 ligand defects. And so because all of these disease-causing mutations are on one single gene, this makes it an ideal candidate for gene editing. And so the reason we pursued this project 5 years ago is because we knew that the CD40 ligand gene is tightly regulated and requires regulated expression. And so we hypothesized that site-specific gene modification, or gene editing, which I presented, in human stem cells will correct X-linked hyper IgM by autologous transplantation. And this can be done using site-specific endonucleases, which are the TALENs and CRISPRs I mentioned, which can target specific DNA sequences for gene modification. And so this is a cartoon depiction of what TALENs look like. And so the way they work is they have two arms, and they can be manufactured in the lab pretty easily. So you can just pretend that the row of letters is the DNA sequence of a small section of CD40 ligand. Let's say we want to target that. Then you can build the TALEN, so the colored circles, you can assemble that in the lab to be specific for CD40 ligand, and that will bind to your goal sequence, and then you have a nuclease called FOC1 that comes together and makes a cut right where you want it to, okay? So there's a DNA binding domain, and then there's a DNA cleavage domain when it comes together. And so what our overall goal is if this is the CD40 ligand gene, let's say this is the very beginning of it, we want to create TALENs that bind to the very beginning of the CD40 ligand gene. These TALENs, when they bind, will create a break in the DNA. And this DNA can be repaired through non-homologous end joining, which we don't want because that creates errors. There's deletions and insertions of DNA. Or if you introduce a TALEN with what we call a homologous donor, so this donor contains human CD40 ligand DNA, that's the corrective template, and it's flanked by these homology arms that parallel the cut site. You could push the cell to undergo homology-directed repair, and trick it into incorporating the normal X-linked or the normal CD40 ligand DNA and keep its expression under control of the endogenous promoter. And when I say that, what it means is now not only have you introduced the normal copy of the CD40 ligand gene, but you're now using the natural on-off switches that are present in the genome. So you don't have to introduce an on switch that's on all the time. Now we're putting it back where it should go, and it'll turn on when it should, and it'll turn off when it shouldn't be on. Okay. And so there's a lot of letters here, but it's basically showing that we tried several TALEN pairs, and it binds to it binds to the start of CD40 ligand right before all the disease-causing mutations start. So if we create TALENs that bind here, it should address all known mutations that are further down. Okay. And the first thing we do is we make sure that these TALENs will cut DNA. So we measured it, and here TALEN 2 happens to cut the best. So we moved forward with this TALEN. And a lot of times when you're first starting something, instead of putting in the normal DNA, you put in something that you can see. So instead of putting normal CD40 ligand DNA, we put in something that shines green when it goes in. So it's called green fluorescent protein. So we took this TALEN and we created a donor that will fluoresce green if it's integrated at the right place. This donor has the homology arms that are very important for homologous recombination. And so when we put the TALENs and this green fluorescent protein donor into cells, in the top you can see that the cells that only get the donor, they don't fluoresce, whereas in the bottom, if they get the TALEN and the green donor, they fluoresce. Okay. And the other really important thing is if we look at these cells that have been gene modified and we look for the green protein in 1 week intervals, we see that this green protein remains there for weeks after we've treated the cells, meaning that we've made a permanent change in the cell's gene. Okay? So the next thing is, how do we know that this green protein is in the right location? And so we kind of measured it indirectly because we know that CD40 ligand expression is low at baseline and it should increase with immune stimulation. And so what we did was we took these cells that were gene modified and we measured it at rest and we stimulated them at increasing intervals to see if the amount of green increases. And it's shown here in the green boxes, but you can see that the amount of green fluorescence increases with immune stimulation. So that was very reassuring to us because it shows us that we've manipulated the gene in a very controlled way. So we're not just putting in, green fluorescent protein anywhere. We're putting it in at CD40 ligand where we want it to occur. And then below, we're just showing that we can kind of play around with different doses and different amounts of donor and TALEN, and we still get a very nice response to different amounts of immune stimulation. And so these were all done in cell lines. So these are immortal cell lines that grow kind of forever in the lab. But what they did for us was they allowed us to establish a foundation that you can manipulate CD40 ligand at the right place, and you can put a gene in there. So initially it was green, but this also suggests that you can now put in the corrective CD40 ligand gene in there. And so what we did was we moved into T cells from one of our patients at UCLA who has X-linked hyper IgM. And so if you remember, I'll just kind of orient you very quickly. So CD40 ligand is shown in the box if it's there. So these are all cells taken from a patient. So you can see that basically there's no cells in the box because this patient has X-linked hyper IgM and does not express CD40 ligand. However, when we treat his cells with the TALEN, it says AAV donor, I should have switched it, but basically a CD40 ligand corrective donor, you can see that when it's at rest, you hardly have any CD40 ligand expression, which is what you want. But then when we stimulate it, like immune stimulated, you can see that now 20% of his cells express CD40 ligand. So this was really exciting to us because we took his cells that have never expressed CD40 ligand and we got it to express correctly. The other, sorry, shown right there. The other very exciting thing is we wanted to see if this CD40 ligand expression was physiologic, meaning does it come to rest and does it get excited and then come to rest and get excited with immune stimulation. And I'll just draw your focus to the light blue and the dark blue points. The light blue, these are all the same cells that have been carried through in culture and have been gene modified for weeks. And you can see that at week 1, if the cells are resting, it expresses around 2% CD40 ligand, which is pretty physiologic. And then when we stimulate it with an immune stimulant, we see that it goes up. And then we let the cells rest for another week. The expression comes down, and we can re-stimulate it. We'll let it rest for another week. It comes down, and we can re-stimulate it. So this is very, very nice because it's showing us that this gene-modified cell is behaving the way it should. The other functional study that you can do to make sure that this CD40 ligand expression is good is not only to look at expression, but to see if it's functioning. And one of the clinical assays that we use is called CD40 binding. So if you remember, CD40 is on a B cell, and so CD40 ligand on the T cell needs to bind to CD40 in order for it to communicate and class switch. And so we can kind of imitate that in the lab, and you can measure the amount of CD40 binding in a test tube. And so these are gene-modified cells, and you can see that these under the blue curve are now the gene-corrected cells that now bind CD40. Okay. So kind of as we were doing this, CRISPR-Cas9 came into the picture, and we wanted to incorporate this because it's, you know, moving forward at, like, so quickly. And so the way it works is it's quite similar to TALENs. It binds DNA and it cuts it. The nice thing is that it's a little bit easier to use. So if this is your DNA target, so if this is CD40 ligand, the way it binds is just this short little guide. It's 20 base pairs long, and it just binds to your target sequence and recruits this protein that will come and make a double-stranded break exactly, always at a predictable place, okay? And so we created, this is the same picture, but what I also wanted to show you was that we created several CRISPRs that also cut right at the start of CD40 ligand, you know, because our goal is to be able to treat as many disease-causing mutations as possible. And so you can see that CRISPR 2 and 3 cut the most, and so we moved forward with those CRISPRs. But instead of putting it just into T cells of patients, which is good and it shows correction, but it's not long-lived, you know, because our T cells will eventually die and they need to get replaced. We started working with gene correction in hematopoietic stem cells. So these are the cell at the very top of that hierarchy that I showed that are self-renewing and that are multipotent, giving rise to all the cells in the blood. And so what we did was we take these stem cells, we put them in culture for a couple days so they kind of wake up because they're frozen when we get them. And then we treat them with either TALENs or CRISPRs, and at the same time give them the corrective CD40 ligand DNA donor. And all this happens quite quickly. The correction happens within a day. And a day later, we can either analyze our samples or we can take those corrected stem cells and put them into other studies. And so first we'll look at what the cells look like one day after, just molecularly. The blue dots are cells that are treated with TALEN and the CD40 ligand donor. And the red dots are cells that are treated with CRISPR and the cDNA donor. And this is quantifying the amount of gene insertion at CD40 ligand molecularly. And so you can see that for TALENs, we average around 12% targeted gene integration, and for CRISPRs, we average around 27% targeted gene integration. And we're continuing to do work with both of them, even though TALENs are lower. We think that even 10 to 12% is probably enough to address the disease, because when we have female carriers of the disease and they happen to skew towards kind of using the defective gene, we know that they can be healthy even with expression as low as 10%. So we're still kind of moving forward with these, both of these platforms. And so just to finish up, I'll kind of show you what we're really focusing on now, because we can correct CD40 ligand in stem cells in a dish like every day. It's now become kind of a daily thing. But what we want to know now is, are we damaging those stem cells? Can those stem cells be transplanted into somebody, like first a mouse, but can they give rise to all of the normal T cells and B cells and NK cells, all those cells, and also self-renew? So is it everlasting? Because we would not want to do this, transplant it into a patient, and then have the stem cells die eventually, right? That's not curative. And so there's two main things that we've been working on to look at the long-term differentiation potential of these corrected stem cells. The first one is actually a very new technology that was developed at UCLA by one of our collaborators. This work was just published last month. But if you remember, Maite talked about T cells. So T cells are what is defective in CD40 ligand. And when those T cells are produced in the bone marrow and they come out into the blood, they're not functional in the blood initially. They have to go to the thymus. It's like University of T cells. So they have to go to the thymus and they have to get selected and mature, and then they come out and then they can do what they need to do. So what people have been able to do is create an artificial thymus in a dish so that I can take these gene-corrected stem cells and I put them into this artificial thymus system, and I keep them in culture for 3 months, and I make sure that those corrected stem cells can now give rise to T cells that can grow out of this artificial thymus. So that's work that we've been working on and has been very exciting. The other thing that we've been working on is kind of along the same lines, taking those corrected stem cells, transplanting them into mice, and then 3 months later looking to see, do those stem cells engraft? Do they produce normal T cells like they should? And how much gene correction is maintained in those transplanted cells? So this is kind of all the kind of really ongoing and kind of exciting work that is going on now. And so I was asked to give just a short list of current challenges, things that we're trying to work around. And I think these are things that will that can be overcome because the field of gene therapy itself is progressing so quickly. So these aren't just things that I'm working on, these are things that the entire field are working on. So one is decreasing toxicity to the stem cells from gene therapy. So, if you remember, I have to get the CRISPR and the talons and the donor in. And the way we do that is we give the stem cell a little shock, like a little thing of electricity to let the talon or CRISPR in. And that little burst of electricity is not great. You probably wouldn't like it yourself. And so it does kill some of the stem cells. And so, that's something that we're trying to see if we can work around or looking at conditions to ameliorate that. The other thing is demonstrating efficacy in vivo, meaning we can do it in vitro in a dish, but does it translate in the mouse or in these long-term thymic cultures? Funding and resources always, because we're always looking to continue to fund this project. And then the last thing is bone marrow and blood samples from affected patients with X-linked hyper-IgM. So I know some people sitting here have donated blood samples to me, which has been enormously helpful because the goal is to correct all the mutations. And so I need to test this in all blood samples from various people with various mutations. And the last thing is bone marrow. So I haven't touched any bone marrow from X-linked hyper-IgM because they're so precious. I've started collecting them because the final step in all this is to correct bone marrow from affected patients, put it into the thymic culture, and also put it into mice and see that they can really engraft and produce normal, healthy T cells. So our long-term goals are to ultimately demonstrate safety and efficacy of site-specific gene therapy for X-linked hyper-IgM. And if we can do this to translate this into a clinical trial, of autologous transplantation and site-specific gene therapy. And so I work with a big group of really very, very smart people who have gotten me to where I am today, and so I always have to thank them. And then I, of course, have to thank my funding sources because they protect my time and keep me in the lab working on this. So I'm happy to answer any questions if you have any.
Address questions to the presenters. Yeah.
Okay.
My son was diagnosed at age 23 with Hyper-IgM Syndrome. The doctors are baffled because the only element that shows up on all of his blood work is this very high IgM level. And when we went to the genealogist and we went for all the testing, they said he's an anomaly. We don't really know why that's the only thing that's different. And they told us that it's going to probably develop into another disease. Is there anything in your background, or you know anything about the one-off situation like that? We still can't find a doctor that's willing to commit or admit. He doesn't present with illness unless he's around a lot of sick people, and then he gets sick immediately, and then he gets really, really, really sick. But for the most part, he's properly taking medicine. He is not on any IVIG product. So I'm just curious to see if any of your research has shown any of these one-off situations on hypertension.
I think the most important thing is to understand what the diagnosis is, right? Because remember, there are many different syndromes that can be associated with elevated IgM, and so not everybody has the really in this era now of molecular diagnosis. You really need to make sure that he doesn't have CD40 ligand, he doesn't have CD40, he doesn't have the NIV, he doesn't have the UNG, he doesn't have the PI3 kinase, he doesn't have the whole list of other conditions that can be associated with elevated levels of IgM. If you think about it, as the B cells are developing, and the first thing they have is that IgM on their cells. And so it's not unusual if there's an abnormalities in the way that T cells talk to the B cells to make them switch, or the B cell itself. Maybe there's a defect in the B cell that even though it's getting the signal, it isn't producing. And we see that in some patients with common variable immune deficiency and other immune deficiency. So my recommendation is if he hasn't seen an immunologist that can have access to genetic testing, that would be the best approach.
I have a question. You've been referring to the donor correction. Do you create that yourself? Is it something you take from a healthy blood sample? How does that come about?
Oh, the corrective donor. Yeah, that's a great question. So the sequence itself is taken from a healthy sequence. So we know what the correct sequence of CD40 ligand should be. But we do make one modification to it before we put it into cells, and it's something that we call codon optimizing. Because when our DNA gets made into protein in our body, there's different things called tRNAs that have to come and pick it up and create the protein. And we know that the tRNAs in our body are different in abundance. And so what we do is we codon optimize it, meaning if I want to make this protein, there's different DNA codes that can make the same protein, but I'm going to choose the code that uses the most abundant tRNA. And the hope for that is that not only will it express the correct protein, but it'll express it better because I'm using what's more abundant, or at least is known to be more abundant in the human body.
And you talked about conditioning. What's, I guess, the difference between the conditioning versus the bone marrow transplant? I know it's like a lighter conditioning or small amounts of chemotherapy, but I guess how does it compare to the conditioning of a traditional stem cell transplant?
Yes, that's also a very good question. So the conditioning for a regular, like the traditional allogeneic transplant, has multiple elements. One is to clear space in the bone marrow, and the other one is to immunosuppress or get rid of the existing immune system, because you don't want what is there to attack the donor cells. For gene therapy, you don't need that second part, because you'll never, or you really shouldn't, attack your own stem cells. So the little bit of conditioning that you're giving is just to create some space in the bone marrow. Because what we know from previous gene therapy trials is that even though you're giving back your own bone marrow, so technically they were there before, is they have no survival advantage when you take them out and correct them. They need a place to go, and so by creating a little bit of space, you increase the rate of engraftment of your gene-corrected cells. So that the dosing is usually less, and they don't require all the immunosuppressives, and usually the hospitalization is much shorter because of that.
So the next phase is to kind of, you did some studies for 21 days, or to see, do these stem cells continue?
Mm-hmm.
Does it continue to express
Yeah.
CD40 ligand?
Uh-huh.
So my question is just basically, how long, I guess, what's the next phase of,
Yeah.
how long would you want to test that? How long would you want to see it to say, okay, well, before you do it on a person, I guess?
Right. So right now, the long-term studies that we've been doing are 3 months. And that's kind of, the starting point for mice and also for this thymic organoid system. We are working, so our kind of overall timeline, if we were to envision this whole project, is, in order to get this into a clinical trial phase, we need to meet with the FDA, and the most important thing besides showing efficacy is showing safety. And so the whole other half of my work that I didn't present here is showing safety of CRISPR-Cas9 and TALENs, showing that the CRISPRs are cutting where it should and not accidentally cutting somewhere else, showing that that toxicity to the stem cell isn't so bad. So our kind of hope right now, and we recently got a little bit of funding to kind of get us there, is at the end of 2 years from today, to be ready for what we call a pre-IND, pre-investigational new drug meeting with the FDA, to be able to discuss our data, to discuss what safety data we have, what efficacy data we have, and to get their recommendations on what else is needed in order to kind of move into the next phase, which would be clinical trial.
So what can we do to help?
I always say bone marrow samples and blood samples. It's a lot to ask, and I totally am aware of that, but that's what's going to show that this works. I can do this every day, and I have a lot of normal stem cells from normal donors, and I can show that all the time, every day. But no one's going to really believe the efficacy of it until I do it in patient stem cells and put those into mice. That is the kind of the standard of preclinical trial work.
So you also said that you did it in one cell, and I was just thinking about DNA and how does it replicate across the board inside the entire human body? You're going to cut, the CRISPR's going to go in, it's going to cut one part.
Yeah.
How does it replicate, I guess?
Yeah, so it's the natural biology of a stem cell. So the definition of a stem cell is that it gives rise. So one stem cell will give rise to many red blood cells, many T cells, many B cells.
Right.
And so if you've corrected that, that's the multipotent. Right. But stem cells are also self-renewing. Right. So they will also, when needed, divide into a second one or divide into a third one on its own.
The only thing I was wondering is, there's other multipotents in there.
Yes.
How do they get changed?
So they won't. They won't.
They don't.
Yeah. And yeah, that's a great question.
And then how does that affect the immune system?
Right.
There's
So
only like X number of total multipotent cells, but only Y number has been
Yeah.
cut, correct it, and then how does that affect the immune system?
Yeah, so that kind of ties into what we were thinking, our kind of tentative cutoff right now is, can we try to correct 10% of the stem cells? Because we think that if because each stem cell gives rise to a lot of T cells. So, and we know that if you have maybe 10%, and Maite can correct me if I'm wrong, but we think that if we have 10% normal T cells, that you could have a pretty functional immune system. So imagine if you were to correct 10% of the stem cells, that may produce more than 10% corrected T cells.